Okay, I am Babu, Professor and Head of the Department of Biomedical Engineering. In this class, I am going to explain about the internal electrodes. Internal electrodes. Okay. Last slide we have seen the types of electrode. In that types of electrode, we have seen the surface electrode, the different kinds of the surface electrode, and what are the characteristics of the surface electrode. Okay, now in this slide we can go for the internal electrodes. So these electrodes detect biopotential within our body. That's why it is called internal electrode. Okay, these internal electrodes, percutaneous electrodes, that is, electrode itself or the lead wire crosses the skin. We are inserting the electrode into the skin so that it is going below the skin. That is percutaneous electrode. Next one is entirely internal electrode. So entirely internal electrode is the connection is to an implanted electronic circuit such as a radio telemetry transmitter. The electrode is entirely available within the body. It is called entirely internal electrode. Then no limitation due to electrolyte skin interface. Okay, in surface electrode, we are placing the electrode on the skin surface. So there is a limitation of the electrolyte skin interface. As internal electrode, it is going into the skin, below the skin, so that no limitation due to the electrolyte skin interface. Next, electrode behaves in the way dictated entirely by the electrolyte interface. So the electrode behaves like electrolyte interface. No electrolyte gel is required to maintain this interface because extracellular fluid is present. So as it is internal electrode, it is available within the body. So no electrolyte gel is required to apply on the surface of the skin. So this is the internal electrode. Okay. So this internal electrode, insulated needle electrode, coaxial needle electrode. So this internal electrode, the types is insulated needle electrode, and the next one is coaxial needle electrode. So this diagram explains about this insulated electrode and coaxial electrode. Next one is multiple electrode in a single needle. Single needle we are having more than one electrode. That is bipolar coaxial electrode. So these are the diagram which is explaining about this A, B, C, D, E, like that we are having the A, B, C, D, E, F. There are the different kinds of internal electrode. Okay, multiple electrodes in a single needle, that is bipolar coaxial electrode, chronic recordings using percutaneous wire electrode. The diagram D explains about the fine wire electrode connected to hypodermic needle before being inserted. Before being inserted, a fine wire is inserted into the hypodermic needle. Then E, the cross-sectional view of skin and the muscle showing fine wire electrode in place. Okay, this is the fine wire is placed in the internally within the muscle or below the skin. Then F explains about the cross-sectional view of skin and the muscle showing coiled fine wire electrode in place. The coiled fine wire electrode is placed in place. So these are some types of the internal electrode. Okay, next we can go for the micro electrode, surface electrode, needle electrode, or you can say internal electrode, and second one is micro electrode. So this micro electrode, the size is very small, and we are going to do experiment with the cellular level. That's why it is called micro electrode. Okay, first, why we need micro electrode? So the micro electrode measure potential differences across cell membrane, resting potential, action potential. These are the potential measured in cellular level. So like this measurement we are using the micro electrode. So the micro electrode measures the potential difference across the cell membrane. Okay. Requirements: small enough to be placed into the cell. It is size is equal to very small. It can be placed into the cell. Next one is not only small, it is strong enough to penetrate the cell membrane. Not only small, it is very strong, enoughly strong to penetrate the cell membrane. Typical tip diameter is equal to 0.05 to 10 microns. So it is in the order of the micron level. Next one is types. So in microelectrode we are having the solid metal, that is tungsten microelectrode, supported metal, that is metal contained within or outside the glass needle, then glass micropipette electrode, that is with AgCl electrode metal. So these are the types of the microelectrode. In that electrode, first one is metal microelectrode, made up of the metal, that's why it is called metal microelectrode. The diagram explains about the metal microelectrode. This is the lead wire, this is the micron level tip, okay. This is the supporting shaft, there is an insulation, and within that we are having the metal. The metal is act as a shaft. So this is end of this we are having a micro level tip of metal. Okay, extracellular recording. This is used for the extracellular recording. That is typically in brain, where you are interested in recording the firing of neurons. Okay, generally we are studying about the EEG, that is spikes, neurons or firing of neurons are generally is called spikes. So this kind of measurements we are using the extracellular recording. Use metal electrode plus insulation goes to high impedance amplifier, negative capacitance amplifier. So here we are having a metal shaft and it is covered with the insulation. Okay. So the tip is connected with your cellular level, that is inside the body. Okay. Next one is metal supported microelectrode. Okay, inside we are having the metal and it is we are having the glass as insulator. Here inside we are having glass and outside we are having the metal film that is also coated with insulation. This is the tip of the two types of electrode. So the first diagram, first figure explains about the metal inside glass electrode. It is also microelectrode. Okay. Outside we are having glass surface, inside we are having metal. So this type of microelectrode is called metal inside glass microelectrode. Next one is glass inside metal microelectrode. So the tip is made up of the glass. It is covered with the metal film and covered with insulation. So glass inside, outside we are having metal. So glass inside metal microelectrode. Then glass micropipette. Okay, a glass micropipette electrode filled with an electrolyte solution. So this is the electrolyte solution, glass pipette microelectrode. This is filled with the electrolyte solution. That is a section of fine bore glass capillary. This is the fine bore glass capillary. Section B, capillary narrowed through the heating and stretching. So in this fine bore, okay, the center we are doing by heating the process, we are making into narrow. Okay, after that finally we are cutting into this kind of structure, final structure of glass pipette micro electrode. So like this we are finally making this fine bore glass capillary, and we inserted the metal micro electrode at the center. This is the lead wire for connection, this is the tip. Okay, this is filled with the intracellular fluid, that is potassium chloride, and this is the cap. This is the structure of the micro electrode. Generally see, okay, this is the diagram. See, intracellular recording, typically for recording from cells such as cardiac myocyte, need high impedance amplifier for this negative capacitance amplifier. Okay, next one is motion artifact. Motion artifact is occurred due to the motion of the electrode. This is the motion artifact. Okay, in this motion artifact, in addition to the offset potential, the electrolyte gel skin potential can also cause motion artifact if it varies with the movement to the electrode. Variations of this potential need to represent a major source of motion artifact in silver chloride skin electrode. This artifact can be significantly reduced. This error can be significantly reduced when the stratum corneum is removed by the mechanical abrasion with a fine abrasive vapor. This method also helps to reduce the epidermal component of the skin impedance. However, the removal of the body's outer protective barrier makes the region of skin more susceptible to irritation from the electrolyte gel. If you apply the electrolyte gel, because the outer surface is removed, it will go for the more irritation. Stretching the skin changes the skin potential. Okay, stretching the skin changes the skin potential by 5 to 10 millivolt, and this change appears as motion artifact. Okay, measurement with two electrodes. So this already we have seen in the last class. So one electrode is another electrode. One electrode is acting as reference electrode, and another electrode is acting as the active electrode. This is immersed in the electrolyte. So the potential developed in between the two electrodes is called offset potential. Okay, this already we have explained. Okay. Up to this we have explained about the needle electrode and the micro electrode. Thank you.